So we start with the third unit of the syllabus, that is biosensors. In this, we are going to study what is a biosensor, what are the benefits of biosensors, okay, types of biosensors. Then we'll talk of biochemical sensors. We'll be talking of chemical potential and equilibrium. Some examples we'll be talking of pH electrode, ion sensitive electrode, etc. All these things we are going to talk about in this particular unit. So let us understand first what is a biosensor. So as the name suggests, biosensors, they are analytical devices that convert biological response into an electrical signal. Now, basically, what you need to understand is biosensor is actually not just one device. Okay, like for example, if you say piezoelectric sensor is one crystal, it's one device. But biosensor, when I say biosensor, is actually not any single device. It is actually a combination of or a setup of devices which can convert a biological response into an electrical signal. Now, biological response might be some enzyme action. Biological response might be, certain maybe sweating, that is also a biological response, or maybe oxidation, blood oxidation, like we say, that is also a biological response. So when we design a certain sensor, analytical device in such a way that it can capture that biological response and convert it into an electrical signal, we call it a biosensors. Okay, the term biological process can be any biological element or material. It might be enzymes, tissues, okay, or microorganisms, cells, acids, etc. So basically, any biological process taking place in your body gives rise to it might produce certain enzymes, it might produce certain acids. Okay, these are types of responses that a human body can give. So when we convert this response, we capture this response and we convert it into an electrical signal, we call it a biosensor. Okay, so biosensor is actually a combination of biological sensing element and a transducer which converts the data into electrical signal. So biological sensing element, what it means that, for example, if it has some enzymes, so first thing that we need is which can detect the production of that enzyme, and then we need a transducer which can actually detect that detection that has taken place. It should be converted into some electrical form. So basically a biological sensing element, it is attached with a transducer, and that can give you an electrical signal. You can see in this top figure, you have a sensory layer, analyte. This you can say these are basically it might be certain enzymes, might be acids which are produced from the human body in response to some action taking place. Okay, this sensory layer that you see over here, it comes in contact with this analyte, it reacts, and this transducer is attached with it, which actually in response to this reaction, it will actually generate an electrical signal, and that will be propagated into an electronic circuitry. So biosensor is a combination of biological sensing element and a transducer which converts the data into electrical signals. Additionally, there will be an electronic circuit which consists of a signal conditioning unit, a processor, microcontroller, and a display unit. So basically, a complete biosensor will be having a biological material, that is a biological sensitive element. It will come in contact, some reaction takes place, that goes to the transducer. Transducer generates a corresponding electrical signal. That electrical signal goes into the electronic circuitry where it is suitably conditioned. Condition means you might want to amplify it, you might want to perform certain actions on that, and then you have a display device where you display it in such a form that it is readable or it is understandable by a simple layman, right? So this whole structure together actually forms a biosensor. Okay, the desired biological material, like in the here you can see the input is coming from biological material. This biological material usually is in the form of enzymes. Okay, that means this biological material is or enzymes. These are produced in the body in response to some biological action. Then it is it is sensed with the help of a biologically sensitive element, and then there is a transducer attached to it which converts it into a corresponding electrical signal. So you can say that there is a biological element. Okay, from that element there comes a biological response. From that biological response there comes an electrical response, and that electrical response is actually measured, and it is then displayed in a suitable form. Let's take one example. Now, glucometer is actually a very, very common biosensor. Diabetes is something which we look around. If we look around ourselves, diabetes is something which we find very common. Okay, so glucometer is a type of biosensor which helps us in measuring blood glucose, right? So, diabetes, it is a disease characterized by the levels of glucose in the blood. Now, excretion of glucose or production of glucose in the blood is actually a response. It is a biological response. Whatever is taking place in your body, digestion, this, that, insulin is acting, and all that is happening is giving rise to some amount of glucose in your blood. So that glucose that is there in your blood is actually a biological response. That biological response we want to measure. Okay, so, regularly checking the blood glucose levels is very important for the diabetic patients. Glucometers, they are type of biosensors which measure the concentration of glucose in the blood. So, usually now you can see the figure. This is what actually it looks like. Usually, they consist of a test strip which collect a small sample of blood to analyze the glucose levels. This particular sensor implements the electroenzymatic approach, that is oxidation of glucose. The test strip considers or consists of a trigger electrode and a reference electrode. When the blood is placed on the test strip, a simple chemical reaction takes place and an electrical current is generated, which is directly proportional to the concentration of glucose. So basically over here, if you see in this small chip, they have, trigger electrode. Trigger electrode is something you can say it works like a battery. Okay, then you have a working electrode and a reference electrode. So this is basically, you must have studied electrolysis in your preliminary classes, something similar to that. That when a electrolyte and a metal comes in contact with each other, some reaction, redox and oxidation reactions takes place, and then current starts flowing, and you attach it to a reference electrode, and you can measure the voltage that is produced, and that voltage is actually proportional to the concentration of that particular metal or the ions in the solution, right? So similar thing is happening over here on this test strip. This is actually a working electrode. When you place the blood sample on that, oxidation of glucose takes place over here. This is called electroenzymatic approach. That means with the help of an electrical electrolysis kind of a thing, you are actually trying to judge what is the amount of glucose present in the blood, right? So when you place the blood sample on this, the working electrode is there. The trigger electrode actually supplies a battery. Electrolysis starts. Okay, and there is the oxidation of glucose takes place, and a corresponding voltage is produced, which is proportional to the concentration of glucose in the blood sample. So this, you can see, this is one biosensor. Okay, now this has certain electrodes. This electrode system that you are seeing over here is actually working like a transducer, which is converting your chemical energy into a electrical energy. Okay, and the working electrode that you see over here, that small strip is actually your biologically sensitive element. Okay, so this is one example of biosensor. This is what biosensors actually look like, and this is how they work. Okay, then we have very different types of biosensors. Very other, they might be enzyme based, they might be tissue based. Okay, DNA based, piezoelectric based biosensors. A lot of the various types of biosensors are there. Enzyme biosensor, they have been devised on mobilization of methods. It is adsorption of enzymes, okay, ionic bonding, covalent bonding, etc. So basically, enzyme based is simple. That is adsorption or absorption of enzyme is taking place. That is enzyme based biosensors. Immunosensors were established on the fact that the antibodies have a high affinity towards their respective antigens. That is, antibodies, they specifically bind to pathogens or toxins or interact with components of the host's immune system. So basically, you can say immunosensor is when you test whether the antibodies are present within the person or not. Okay, so immunosensor, that means how much a person is immune, that you that sense you get from here. Okay, so basically, if you have that immunity sensors or antibodies present in the blood sample, they will have a tendency of combining with the pathogens immediately. So when you bring the pathogen in contact with the blood sample, if they combine quickly, that means the antibodies are present in your human blood, in your blood sample. That means you already have a defense mechanism towards that particular pathogen. So that is also a kind of a biosensor. Then you have DNA based sensors. They are devised on the property that single strand nucleic acid molecule is able to recognize and bind to its complementary strand in a sample. So DNA detection, that we say, that is basically DNA based biosensors. Okay, then you can have piezoelectric biosensors. They might be quartz crystal microbalance and surface acoustic wave devices. They are basically based on the measurement of changes in resonance frequency of the piezoelectric crystal due to the changes in mass. Okay, so if some changes are taking place, some extra mass is produced, that is when that applies extra pressure on the piezoelectric crystals, it gives you a corresponding voltage. Okay, so we have a very wide, not only in biomedical applications, we have actually a very wide application of biosensors. It is used in drug developments. Okay, it is used in the replacement organs, prosthetics, that we say, it is used in that domain also. In food processing, biosensors are very popularly used, monitoring food authenticity, quality, safety, and all that. Then medical diagnosis, of course, like I said, glucose biosensors, they are the most widely used for diabetes. So that is one example. And then bio warfare. Biosensors, they can, actually be used for military purposes at times of biological attack. Okay, the main motive of this biosensor is to be sensitively and selectively identify the organism posing threat in virtually real time, called biofare agents, namely bacteria, toxins, and all that. So if it is, biosensors can actually be used to detect if there is a spread of some particular bio warfare agents. That in that way also, biosensors, they are very effective. So you see, in the upcoming sessions, we'll be talking of how this works. We'll be talking how ion selective samples are detected, how partial pressures can be judged in a blood sample. This all comes under biosensors. Okay, so biosensor is actually not one device, whereas it is a setup. It can be a combination of any kind of a device which is actually capable of detecting a biological response. Biological response might be an enzyme production, might be some tissue, might be some acid production, something like that. That is a biological response. So if your sensor is capable of detecting that and converting it into a electrical signal, it becomes a biosensor. So it is a combination of a biological sensitive element and a transducer which converts it into an electrical signal. That electrical signal is fed into a signal conditioning unit where amplifications and other modifications are done, and finally it is displayed on your display device in certain comprehensible form. Okay, so what types of biosensors can be, how electrolysis and biochemical sensors we can design, that we'll be talking about in the upcoming sessions.